Welcome to another episode of Tiny Talks Big Ideas podcast. Today we're embarking on, well, a truly profound journey, I think. A deep dive into a single discovery that fundamentally transformed how we see life itself. Just imagine for a moment the sheer wonder of it all. You can send off a tiny bit of saliva, and back comes this detailed map of your ancestry, like where your family came from generations ago, all from just a few cells. Or think about forensic teams. They can identify someone from maybe just a single hair left at a crime scene. It really feels like magic sometimes, doesn't it?
Oh, absolutely. It does feel like magic. These applications are, frankly, stunning, almost unbelievable when you really stop and think about how powerful they are. It's kind of amazing.
Well, that sense of wonder, that feeling of magic, that's exactly what we're going to unpack today, because the root of all these incredible things, it lies in a single, groundbreaking, and actually surprisingly short scientific paper, published way back in 1953. And this paper, it wasn't just floating an idea. It claimed to solve one of the deepest mysteries there is, the secret of life itself.
Wow, okay. The secret of life itself. That's a huge claim for one paper. Sets the bar incredibly high, wouldn't you say?
Oh, it really did. But, the paper's title is A Structure for Deoxyribose Nucleic Acid by James Watson and Francis Crick. And looking back, it's almost funny how understated it is. Because what they laid out, that became the absolute foundation for modern biology. It changed everything. Think about where science was in the early 1950s. Okay, so scientists knew DNA, this deoxyribose nucleic acid, was important. They suspected it was the stuff of genes.
The blueprint, right. The instruction manual for life.
Exactly, the instruction manual. But here's the thing. They couldn't read the manual. They had no idea how it was structured.
So they knew what it did, sort of, but not how it did it.
Precisely, and maybe even more critical, they couldn't figure out how this incredibly complex set of instructions could be copied perfectly, flawlessly, every single time a cell divides. How does life pass on its instructions so accurately, generation after generation? That was the big mystery.
It must have been like looking at a black box. You know something amazing is happening inside, but you just can't see the mechanism.
That's a perfect analogy. It was a black box.
Yeah.
And cracking it open, figuring out the structure of DNA, became this intense race, huge stakes. You had some major players involved. Over in America, there was Linus Pauling.
Ah, Pauling.
The giant.
A giant,
Yeah.
exactly. Brilliant chemist, Nobel laureate already. He was definitely the heavy favorite. And he proposed a structure, actually, a triple helix, 3 chains twisted together.
3 chains, okay.
Yeah. And crucially, he put the phosphate groups, which are negatively charged, on the inside of this helix. Bases on the outside.
Phosphates on the inside.
Right.
That sounds potentially problematic, chemically speaking.
You got it. Watson and Crick over in Cambridge, they heard about Pauling's model and apparently realized pretty quickly it couldn't be right. Like you said, cramming all those negatively charged phosphates together on the inside, they'd repel each other like crazy. The molecule would likely just push itself apart.
So Pauling stumbled. Even the giants can miss things. He did. And that stumble kind of opened the door. Meanwhile, you had Rosalind Franklin working at King's College in London. She was a truly brilliant X-ray crystallographer, incredibly meticulous, producing just the best, clearest X-ray diffraction images of DNA anyone had ever seen.
Right, X-ray crystallography. Maybe you should quickly explain that. It's sort of like figuring out the shape of something you can't see directly.
Yeah, go for it.
By shining X-rays through it and analyzing the patterns of the shadows it casts. It's incredibly complex, but those patterns hold clues about the molecule structure. Franklin was a master at getting and interpreting those shadow patterns.
A true master. And her images, especially one known as Photograph 51, were just stunning. It practically screamed helix with this distinctive X-shaped pattern. Crucial evidence.
That X is the classic helical signature, isn't it?
It absolutely is.
Yeah.
Then you have our sort of underdogs, Watson and Crick in Cambridge. Watson, a young American biologist, Crick, a British physicist. They weren't even technically supposed to be focused on DNA structure, but they got obsessed with it. And their approach was different. They were building physical models, like out of wire and metal plates. Kind of like a molecular Lego set, trying to make all the pieces fit based on the known chemical rules and the available data.
A very different approach from Franklin's pure experimental work or Pauling's theoretical calculation.
Exactly. And then through a series of events, and we should acknowledge there's still some historical debate about exactly how this happened. Watson and Crick got to see some of Franklin's unpublished data, including Photograph 51.
Ah, that's the controversial part of the story, the access to her data.
It is. But that data, especially the clarity of Photograph 51, seems to have provided the final, absolutely vital clues they needed to figure out their model. which brings us back to their paper. And the way it starts is just so modest. They write, "We wish to suggest a structure for the salt of deoxyribose nucleic acid."
Wish to suggest. Like, it's just a thought they had.
Right. And then they add this little gem.
Hmm.
This structure has novel features which are of considerable biological interest.
Considerable biological interest. That has to be one of the biggest understatements in scientific history.
Isn't it just incredible? So what was this structure that was merely of considerable biological interest? Well, first off, it was radically different from Pauling's idea. Not 3 chains, but 2. 2 helical chains coiled around a central axis.
The double helix, the icon.
The icon itself, the twisted ladder we all recognize. And crucially, those phosphate sugar backbones, they weren't on the inside like Pauling thought. They formed the outside rails of the ladder.
And putting the backbones on the outside makes perfect sense, doesn't it? It solves that repulsion problem immediately. The negative phosphates are on the exterior, interacting with the watery environment of the cell.
Exactly. Stabilizes the whole thing.
And importantly, it means the bases, the A, T, C, and G, the actual letters of the genetic code, are tucked safely away on the inside, protected.
Yes, forming the rungs of the ladder, perpendicular to the main axis. And it was more elegance, too. The 2 chains run in opposite directions. Anti-parallel is the term, like traffic on a 2-way street.
Which turned out to be super important for how it copies itself later on.
Absolutely. And they figured out the precise dimensions, too. Like, each step up the ladder is 3.4 angstroms. There's a specific twist angle. It repeats every 10 steps. Really precise geometry.
It wasn't just a vague, twisty shape. It was very specific, defined structure.
Very specific. But here's the real kicker. The absolute genius insight. The pairing of the bases across the ladder, the rungs. They realized it wasn't random. To keep the ladder perfectly uniform and with no bulges, no pinching a larger base, a purine like A or G.
Adenine or guanine, yeah.
Always had to pair with a smaller base, a pyrimidine like T or C.
Thymine or cytosine. So big always pairs with small. Keeps the distance between the backbones constant. Neat.
Super neat. Structurally elegant.
Yeah.
But they took it one step further. Working with their models, they found it wasn't just any big with any small. Only specific pairs could form the right number of hydrogen bonds to hold the chains together stably. Adenine A could only pair effectively with thymine.
A with T. Okay.
And guanine G could only pair effectively with cytosine C.
G with C. So that's it. AT and GC. Always.
That was the rule. The fundamental pairing rule.
And the implication there is just huge. It means the 2 strands aren't just parallel, they're complementary. Like you said, like a photograph and it's negative. If you know the sequence of one strand, say, it goes AGGTC, you automatically know the other strand must be
PCCAG. Exactly. You just know it. It's built right into the structure.
Mind-blowing redundancy and information storage.
And that insight led to the paper's legendary and, again, incredibly understated final sentence. It has not escaped our notice that the specific pairing we have postulated immediately suggests the possible copying mechanism for the genetic material.
Boom. There it is. That's the mic drop moment, isn't it? They didn't just show the structure. They basically handed everyone the key to how life copies itself, how heredity works at a molecular level. Just imagine if that double helix unwinds, zips apart right down the middle.
Splitting the base pairs.
Right. Then each of those single strands acts as a perfect template, a mold. Because of that strict ATGC rule, the cell can just grab new matching bases from the surroundings. A new A lines up opposite the T, a G opposite the C.
And so on down the line.
Until, bingo, you've built complete new complementary strand for each of the original strands. You end up with 2 perfect copies of the original DNA molecule.
2 identical double helices where there used to be just one.
It's just so elegant, so simple, yet so powerful. The structure explained the function. That's the holy grail in biology, isn't it?
It really is. Finding structure and function together like that, almost unheard of. And the impact was immediate, like a lightning strike. This structure, the double helix, it was just too elegant. It explained too much experimental data. It made too much sense to be wrong.
It had that ring of truth.
It really did. The scientific community pretty much accepted it instantly. Just threw open the floodgates for biology research.
You used the term black box earlier. It feels like they didn't just open the box, they found the Rosetta Stone inside. Suddenly, the language of genetics, of life, started to become readable.
That's a fantastic analogy, the Rosetta Stone for life. And everything cascaded from there. This discovery is the bedrock of molecular biology as a field. It directly enabled massive undertakings like the Human Genome Project in the 90s and early 2000s.
Yes, mapping out all 3 billion letters of our own genetic code. Unthinkable without knowing the structure first.
Absolutely unthinkable. And think about today. We started with those examples. Your ancestry test relies on comparing tiny variations in that code. Crime scene DNA analysis depends on matching that code precisely. Even things like the recent mRNA vaccines.
Based on manipulating genetic instructions?
Precisely. And looking ahead, these incredible gene editing technologies like CRISPR, the ability to potentially go in and fix faulty genes, cure genetic diseases.
It all stems back to knowing the structure, knowing how the information is physically arranged.
None of it would be remotely possible without Watson and Crick and Franklin figuring out that twisted ladder back in '53.
It really makes you think about the people involved. You mentioned Watson, Crick, and Franklin. How was the recognition handled? Did they all get the credit due for such a monumental discovery?
That's a really important point, and the story's a bit complex there. Watson, Crick, and also Maurice Wilkins, who was Franklin's colleague at King's College and also worked on X-ray diffraction of DNA. They shared the Nobel Prize in Physiology or Medicine in 1962.
Okay, so they were recognized at the highest level.
They were. But Rosalind Franklin, her story is quite poignant. She tragically died of ovarian cancer in 1958 when she was only 37.
So young. And before the Nobel was awarded.
Exactly. And Nobel Prizes aren't awarded posthumously
Yeah.
after someone has died. So she couldn't share in that prize.
Oh, I see.
And for many years afterwards, honestly, her absolutely crucial contributions, especially that critical photograph 51, weren't fully appreciated or acknowledged as widely as they should have been.
That's unfortunate, but sadly not uncommon in the history of science sometimes.
True. But thankfully, that's changed significantly over the years. Historians and scientists now fully recognize just how essential her experimental work was. She's rightly celebrated today as a key figure in the discovery.
That's good to hear. It highlights that the legacy of a discovery like this isn't just the science itself, but also the human story behind it. You know, the collaboration, the competition, the personalities, sometimes the injustices.
Yeah.
It's all part of how science actually gets done.
It really is. And the story isn't over, is it? The impact of discovering the double helix is still unfolding, still growing, maybe even accelerating. We've moved from reading the DNA code, which was revolutionary in itself,
A massive leap.
to now starting to write it, or at least edit it, developing therapies that could potentially correct genetic errors at their source, curing diseases we once thought were incurable.
The potential is just staggering.
It really is. That short, modest paper from 1953 didn't just solve a mystery. It gave us the tools, the fundamental understanding, to begin actively interacting with, maybe even engineering, life itself. Thinking about that transition from reading the book of life to potentially rewriting it. What does that mean for the future? What are the next big questions this opens up?
Well, the ethical questions alone are immense, aren't they? But scientifically, it pushes us to understand gene function, regulation, interaction on a whole new level. It's not just about the sequence anymore, but how it all works together. The story is definitely still being written.
Until next time we meet in another episode of Tiny Talks Big Ideas podcast, stay safe, stay healthy, and stay curious.